This is Assistant Professor of Pharmacy, Soykan Ağar, from the University of Kocaeli Health and Technology University. I will introduce the topic of how AI integrated computational simulations will shape the future of drug discovery. I will begin describing my CV quite a bit right now, and then I'll go on with the details of my studies and the future of drug discovery with AI. I graduated from Koç University, the Chemical and Biological Genetic Engineering program with high honor degree. I went on my studies with Istanbul Technical University in the master and PhD degrees in the area of medicinal chemistry, the in silico drug design and repurposing. I went abroad, worked in various universities in France, Italy, United States, and finally I was at University of British Columbia, where I was a doctoral teaching and research fellow over there, teaching in silico bioorganic chemistry and drug design courses and researching on them. I won a prestigious NSERC project from the Government of Canada. Then I was a postdoc teaching fellow at Eastern University in the Faculty of Pharmacy, where I was teaching in silico drug design and characterization courses. And finally, I became an assistant professor in the Faculty of Pharmacy at Kocaeli Health and Technology University, where I currently European Cooperation Oncological Working Group member and teaching organic chemistry, microbiology, etc. Those kind of courses, where my group is primarily focused on in silico drug design. So I'll begin my presentation with the benefits of AI and simulation drug design. The fundamental concept in AI health sciences, such as pharmacy, medicine, genetic engineering, medicinal biochemistry, and medical biochemistry fields, possess following elements in the near future. The discovery of new drugs, de novo drug design, will be made quite efficiently with AI integrated programs. Currently, simulational programs are being used quite often, but there are new AI modules that are being integrated onto these. So this has begun to shorten these times. And this whole procedure can only be achieved through the combination of advanced knowledge and great grasp of organic chemistry for the drug design. The actual efficacy of a molecule designed with organic chemistry, pharmaceutical chemistry knowledge is now anticipated not only through laboratory experiments, in vitro, in vivo, but also by simulating its interactions with cells beforehand. A pharmacist, pharmaceutical engineer, medical doctor, or genetic engineer with a strong foundation, possessing a profound understanding of organic chemistry and AI usage, can significantly influence the world of pharmaceuticals from now on. In the modern world, the AI technologies, machine learning, computational simulation software, and the organic pharmaceutical chemistry de novo drug design has expanded its variety of tools to battle the spread of cancer or such related diseases in our fields, to a point where it'll be even wiped out from the earth once and for all in the near future. It seems that way. So to be able to achieve such a difficult task, medical doctors, genetic engineers, pharmacists, and such need to upgrade the methodologies of designing and producing the anti-neoplastic drugs, especially in the oncological field, my field. In this manner, rather than the conventional ways of drug production that were taking something around 15 years in FDA, it has begun to shorten to a period of 5 to 6 years with the AI integrated computational software. So in the past, the FDA approvals for each drug, for each molecule that are getting designed, were taking up to 10 to 15 years minimum, with a total amount of $2 to $5 billion. But with the new modern techniques, including AI, machine learning, and simulation studies that are being used to shorten this possible de novo drug design period, by also suppressing the side effects, toxicological effects on them, and increasing the efficiency and indications of these drugs. This has been made possible something around 5 to 6 years. So my current group, within my current group, I have a postdoc from Istanbul Technical University, Barbaros Akurt. I have 2 master degree graduate assistants and a lot of undergraduate assistants at COSTU, Kocaeli Health and Technology University, where we are only focusing on AI integrated drug designs. So in terms of further discussing the benefits of AI and simulation drug design, I should mention that the in silico AI studies allow many probabilities of molecule bindings to be tested before going into further wet lab experiments. Thus, it saves time for finding the right drug molecule, which time is equal to money, and it means saving loss of lives, especially in diseases such as cancer. So it has a significant role in that term. Such in silico simulation centers like mine take less physical space compared to the ones in wet labs. It's easy to be set up from scratch, and the cost of all these maintenance only comes from supercomputers. You don't buy any chemicals, you don't buy any biological kit purchases. There's nearly a zero cost, only the maintenance of supercomputers and the electricity. So when the in silico drug simulations decrease the chemical and pharmaceutical synthesis expenditures, de novo drug design can spend hundreds of chemical structures to find out the correct drug formula fitting for the right indication to the target, which we call receptor molecule within the in silico terminology, inhibiting the diseases or symptoms. Big data analysis with AI software allows the correct chemical functional groups to be found. While doing so, high yield drugs will be able to be synthesized faster than ever before, while being environmentally green with the high precision target indications. My specific academic area is on the drug discovery of binding mechanisms of de novo drugs that can cause apoptosis for cancer cells. In this manner, gene silencing factors, nucleotide regioselectivity factors, and DNA domain selector factors, depending on the alteration of functional groups on the designed drugs, are untouched fields that should be revealed. So this is a pretty new area that scientists like me should be the pioneers in this field, so that any new research on this area is very valuable. Therefore, since the spread of cancer in humans increased day by day quite rapidly, the anti-neoplastic drug design has become one of the major research areas in computational and medicinal chemistry. In the field of medicinal chemistry and oncological research, intertwined with artificial intelligence integration, the pursuit of innovative organic pharmaceutical drug design has emerged as a cornerstone strategy. This extends beyond exclusively targeting DNAs and encompasses a comprehensive approach toward the intricate pathways, as well as the molecular targets of RNAs, mRNAs, regulatory RNAs, and proteins. In the current landscape, pharmaceutical companies find themselves in the midst of an area marked by the excessive production of chemotherapeutic drugs, anti-neoplastic drugs, predominantly composed of non-targeting molecules. More academic collaborations can solve this issue, where targeted molecules need to be only synthesized and applied to the patients in clinics. Non-targeted molecules are not quite specific, and they have high toxicity values. And therefore, pharmaceutical companies tend to not use them in this era. Yet within the annals of scientific history, certain cancer-suppressing drugs designed to target specific elements have been synthesized and utilized, albeit with elusive underlying mechanisms. The pharmaceutical landscape is adorned with FDA approval anti-neoplastic drugs, but their mechanisms remain in mystery. Unlocking the cell biological intricacies and effects of these drugs could potentially catapult their capabilities to unprecedented heights, delving into the uncharted territory of understanding which molecular domains engage with cancerous cell DNA, elucidating the binding modes, and comprehending the stabilization mechanisms can provide invaluable insights for future drug designs. Central to this exploration are chemotherapeutic DNA damager agents in the field of oncology, which are compact cytostatic molecules that frequently bind and unpair the double-stranded DNA. These are the what we call DNA intercalators. They get into the stacks of DNA helical strands and destroy them completely, separate them completely. And we're trying to make them focus onto the specifically just the cancer's cell DNAs, not the healthy cells. And these have been achieved in our studies, which I will mention later on. This binding induces modifications in structural and nanomechanical properties, disrupting the proliferation process of cancer cells and triggering apoptosis. Cytotoxic drugs, revered for their varied interference with cell division, seek to inhibit the process with the ultimate goal of eliminating cancer within the host. So the trajectory of pharmaceutical drug discovery, in especially the oncological arena, has steadily evolved quite rapidly, I must say, aiming to silence cancerous DNAs by either inhibiting their protein expression or irreversibly dismantling their structure completely. In the realm of DNA-targeted drug designs, this lofty objective is realized through gene silencing, employing groove binding methodologies, shutting down DNA via external phosphodiester binding over this area, or obliterating the helical strands through the middle of the structure, disrupting the hydrogen bonds within the nucleotides, and completely dismantling the helical strands is the key in the design of these drugs, intercalator drugs. The theoretical foundations of these approaches necessitate empirical validation through multispectroscopic experimental methodologies, such as the use of FTIR, NMR, absorption spectroscopy, etc., for the in vitro parts of the experiments after our in silico drug designs are sure of the structure of the molecule of the drug. And later on, the cell line inhibitions come into play, where we try to recognize whether this molecule of interest that we design can inhibit the cancerous cell line or not. So, and after that procedure, the part where in vivo, ex vivo, and human phase clinical trials come into play. This imperative step bridges the gap between the theoretical computations and practical applications, unraveling the hidden mechanisms of drugs and providing a comprehensive understanding of their capabilities. As part of this ongoing pursuit, pharmaceutical chemical databases become meticulously curated to encapsulate the properties of studied drugs. This virtually scanned molecules create a huge library that have affinities towards the target of molecules of interest. So we begin with the virtual screening of vast amounts of molecules. We use doc.ai, such a program that is integrated with our computational docking programs. We reduce the number of molecules. We test the scores of dockings. Then we go on with the clustering analysis. And then when we choose the right molecules among them that have the correct clusterings and good docking results, do good docking scores with minimum of delta G kilocalories per mole values, we apply MD scoring, which we go into the molecular dynamics part, where we evaluate the MMGBSA scores. And then after that procedure, there's a manual filtering using our oncological chemistry knowledge, where we evaluate those molecules and find the most right-fitting drug-like molecules that have the most indication values with the least toxicological effects. So the inception of new chemical structure on paper, devoid of laboratory synthesis or experimental validations, prompts a need for predictive assessments concerning its indications, toxicity, and binding mechanisms on the target molecule with specific region selectivity. The ability to foresee the preferred functional groups of target molecules and their strong bonds, whether it's a hydrogen bonding or covalent bonding, depends on the mechanism that you want. If you want a reversible mechanism, it'll be hydrogen bonding. If it's an irreversible bond, you may choose to be working with covalent bonding. The ability to foresee these preferred functional groups of target molecules, and as well as the prediction of repeating amino acids or nucleotides and their frequency, this plays a pivotal role in shaping the trajectory of drug design and development. The intricate intersection of medicinal chemistry, oncological research, and artificial integrated intelligence heralds a new era of pharmaceutical innovation. The necessity of advanced organic chemistry is quite crucial in this manner, since the pursuit of targeted drug design is grounded in a deep understanding of the organic chemistry of molecular interactions. So one that will study this area, this in silico AI integrated in silico area, should very well know and has a very well knowledge in advanced organic chemistry. This promises to revolutionize the cancer or any other type of disease treatment paradigms, ushering in an era where drugs are not only efficacious, but also precisely tailored to combat the complexities of the disease. So let's go on our presentation with the AI innovations in drug design, what our group has achieved in that manner, which I'll come in a bit of later on slides. So this area is like a key and a lock. The key is called a ligand molecule, a molecule that will make the attack onto the target, which will do the suppression of the receptor, the target molecule, so that the disease can be halted or inhibited. Sometimes in oncology, we prefer halting, suppressing, stopping a metastasis, or we may choose the suppress the oncological unit, the target molecule completely, so that we can once and for all shut down the cancerous cell and cause it to go into an apoptosis. And the lock mechanism, the lock itself is like the target molecule, which is involved in a significant biological process for the disease. So the organic chemistry and medicinal chemistry used in drug design are crucial to find the correct leads. Being good at drug-like affinity tendency knowledge is one thing from the perspective of a researcher, but to be able to find the significant needles in the haystacks, molecular dynamics studies have gone under AI tools as well. Molecular dynamics and its post-molecular dynamic tools already provide great estimations in simulations and enough tools to find precise drug-like molecules for testing. However, to increase the pace of molecule scanning, this virtual molecule scanning and affinity estimation, the AI software is very, very helpful in this manner. You can see some of the programs that we choose to use. With such good supercomputers at hand, virtually scanning new molecules utilizing integrated AI and new post-molecular dynamics methods, one can find accurate and precise drug-like molecules easily within minutes or hours per each round for each molecule. Since I became an assistant professor at Kocaeli Health and Technology University, as soon as I've gotten my tenure within this university in the Faculty of Pharmacy, within like 6 or 7 weeks, as soon as the supercomputers were provided to me for my in silico lab, I was able to find tens of new efficient molecules that can suppress pancreatic cancer, prostate cancer, head, neck, oral cancer, fatty liver disease, NASH disease, etc. Thus, I have created a massive library of virtual molecules, just like 1.5 to 2 month. I mean, this area is hugely expanding in this manner. So with my assistance, we started writing many research papers and applied for patents for each molecule that we find that had efficient binding and suppression capabilities. And we've written methodologies immediately for patenting this as well, for patent ownerships as well, which they were all accepted by prestigious journals and the Turkish Patent Office. Then our group was accepted to Cost Action, European Cooperation in Science and Technology in the Oncological Research Working Group from Turkey. Now we have a wide network with Europe, where we're discussing these issues with them and adding valuable information for the sake of Turkey. All of these new discoveries had nearly 90 to 95 accuracy compared to the cell line wet lab studies. You may ask why? Because computational simulation drug design software has come to a great point where it can estimate the reality of the biological systems very efficiently with the most updated software and force field computations. What this does mean is that we're at the level where we can simulate the whole nuclei, the whole elements of a nucleus. I'm assuming you're working on DNA. So instead of just trying to suppress a specific protein, a specific RNA segment, we're at the level where we can study the entrance of the drug into the nucleus through the nuclear, the membrane of the nucleus, how it can pass, and after it passes, how it will evolve and become metabolized, and how it will interact with the gene, which domain of the gene it'll select, what's its regioselectivity gonna be, and does it have any regioselectivity for a specific nucleotide? All of these questions are now being answered, and we're working on these simulations. And this whole universal simulation, this whole grid, the arena of the research is getting larger and larger, so that we're finding more accurate results. And maybe the Earth will be wiped out from all these diseases when we'll be able to study on the whole cell, or maybe the intracellular matrix, and maybe we'll be able to add veins, intracellular matrix, and the cells, so that we can simulate the whole body maybe in the future, so that without even going into the in vitro and in vivo lab, we'll be able to get all these results with 95 accuracy for the whole body, maybe in the future, so that such advancement in the supercomputers are now providing us invaluable data. And this is a huge, tremendous advancement in the technology right now. So any information that are computing via simulations with molecular ducking, molecular dynamics, and AI software, decreasing the time and the money spent in these other experimental parts, in vitro, in vivo, human phase, clinical trials, etc. So especially very high accurate drug molecules computed in the in silico stages shorten this production of any molecule of interest that is highly specific to its target. And this time period is being shortened and shortened by day drastically. So in terms of simply explaining what a hit discovery is, I'll give this slide to you. I'll give a brief explanation regarding this issue. In a hit discovery, which is simply a process of finding an efficient molecule that can suppress the disease cell targets molecule from billions of molecules, AI-based virtual screening begins at this area, where until a good ducking result can be obtained, which there is a filter of eliminating the other molecules using the ND molecular dynamics tools integrated with AI. Then AI goes on predicting results by altering the functional groups of those good results. It alters the molecules functional groups into each other using thousands of functional groups and makes a bombardment onto the target molecule until it gets the nearly the perfect results for the finding the correct structure, and reruns its estimation tests by iterations so that it finds the perfect molecule. If all goes well with our manual molecular dynamics tests as well, a great hit molecule that we call is yielded, and it's ready for further organic synthesis, in vitro molecular characterizations, and go into in vivo and human-based clinical trials. So it becomes a huge part of this new branch, I must call in this manner. So to conclude our presentation, I must say that the future of drug discovery, especially in the fields like oncological research like mine, lies in precise targeting of receptor molecules, cancerous molecules, whatever their origins are. Speaking of a specific example, in oncological AI simulations, there is no more cancer types classifications. We used to say breast cancer or head neck cancer, prostate cancer, but this type of classification is now getting changed. Says whatever the target molecule's origin is, whatever its specific biological pathway is, that's our own solely the concern for us. The tissue or the organ that is origin is not the classification method right now. The whole science is changing with this AI integrated in silico studies right now. And this emphasizes the great change and the significance of this area, as I've written onto your slide. Our ongoing studies have revealed promising insights into various drugs, contributing valuable data to our organic pharmaceutical chemistry database, particularly in understanding roles of functional groups. By scrutinizing the affinity, the binding score, the mode of binding, with which mechanism it binds to which nucleotide or which amino acid, and the nucleotide region selectivity, with nucleotide it chooses in terms of chemical structure of different functional groups in drugs, we were able to draw meaningful comparisons and contrasts. This analysis involves examining the overlapping outcomes from computational simulations, AI software, and multispectroscopic analyses. Through this comprehensive approach, it becomes possible to anticipate both the new effects and repurposing effects of de novo designed drugs. The goal is always to create medications that are not only efficient and enduring, long-lasting, but also have reduced toxicity and a specific region selective focus on their target. Thank you.